Alright, so we're going to talk about the central dogma of biology, specifically the first step, which is transcription. Transcription is the generation of an RNA molecule from a DNA template. So we have DNA to RNA. So all nucleic acids, both DNA and RNA, have directionality, going from a 5' to a 3' direction. DNA is double-stranded, but goes in the opposite direction, or anti-parallel to each other, and forms a helix, while RNA is a single-stranded molecule, which still has directionality. Whenever nucleotides pair up, they rely on this directionality to create valid base pairs between A's and T's, C's and G's, or A's and U's. So the DNA has two strands, like I mentioned, which can be the coding strand or the template strand. The template strand is the strand that is used as a template to create the RNA through base pairing. The coding strand has the same genetic code as the RNA strand because of base pairing. However, the coding strand does not interact with the RNA polymerase enzyme as the RNA is being generated. So after transcription from the template strand, we have an mRNA molecule. So now we're going to do an example. So we have an mRNA strand here given 5' to 3'. Now if the 5 and 3 were not labeled, we would assume for most nucleotide sequences that it is in the 5' to 3'. However, here it's labeled, so we know it for sure. So if we were to determine the template DNA strand that generated this mRNA strand, or this tDNA, T for template, we get 3'. So A base pairs with T, U base pairs with A, and so forth and so on. So if we were to create this, we would have this as a template DNA strand. So then if we wanted to find what the coding DNA strand is, or cDNA, that would be given in a 5' here, which we can find from either base pairing with the template DNA, or we know that the coding strand has the same sequence as the mRNA strand due to the polymerization reaction that makes RNA by base pairing with DNA. So therefore, we can take the mRNA strand 5' to 3' and write it the same by replacing the U's with T's. Because T's are in DNA, where U's are in mRNA. And then if we wanted to, we could take each codon to continue the central dogma and create the proteins.